The field of synthetic biology is constantly evolving and producing new and exciting breakthroughs. Recently, researchers have engineered E. coli bacteria to produce proteins for use in vaccine production, which is faster and cheaper than traditional methods. Another exciting development is the use of yeast cells engineered through synthetic biology to produce biofuels from plant-based materials, making them a promising option for sustainable energy production. In addition, synthetic biology is being used to revolutionize the way we fight diseases, such as by engineering bacteria to produce drugs that can treat cancer and autoimmune diseases. Another application of synthetic biology is in bioremediation, where researchers have created artificial cells capable of breaking down toxic chemicals. Synthetic biology is also being used to capture carbon dioxide from the atmosphere and store it in a stable form, helping to mitigate the impacts of climate change. These are just a few of the many exciting advances in the field of synthetic biology. If it's exciting enough, let's see how it was discovered. Once upon a time, in the early 21st century, a group of scientists made a groundbreaking discovery in the field of biology. They discovered that they could manipulate and control the behavior of individual cells by programming them with synthetic biological components. In 1973, the first recombinant DNA molecule was created by the molecular biologist Herbert Boyer and the geneticist Stanley Cohen. This was a significant breakthrough and marked the beginning of a new era in molecular biology. With this new technology, scientists were able to create new strains of bacteria and yeast that could produce human hormones and insulin. In 1976, Paul Berg, a biochemist at Stanford University, became the first person to genetically modify a virus. This was another major milestone in the development of recombinant DNA technology and set the stage for the creation of novel biological systems. In the following years, scientists made significant progress in the field of recombinant DNA technology. They developed new techniques for cutting and splicing DNA, and for transferring genes between organisms. This allowed scientists to explore the intricacies of life at the molecular level, and opened up new possibilities for the study of genetics and biology. By the end of the 1970s, recombinant DNA technology had become widely adopted by scientists in different fields. The technology was being used to study the fundamental processes of life, and to explore the potential applications of genetic engineering. This marked the beginning of a new era in biology, and laid the foundation for the development of synthetic biology. One of the first applications of synthetic biology was in the creation of biofuels. Scientists discovered that they could program yeast cells to ferment sugars into ethanol, a process that was previously only possible with crude oil. This new biofuel was cleaner and more sustainable than traditional fossil fuels, and it quickly gained widespread popularity. As the field of synthetic biology matured, scientists began to push the boundaries of what was possible. They created bacteria that could detect and clean up oil spills, plants that could purify contaminated water, and even bacteria that could produce life-saving drugs. The discovery of synthetic biology also had a profound impact on the field of medicine. Scientists were able to design and engineer cells that could seek out and destroy cancer cells, leading to a new era of personalized medicine. Despite its many benefits, synthetic biology was not without controversy. Some people raised concerns about the safety and ethics of programming living organisms. They worried that the technology could be misused, or that synthetic organisms could escape into the environment and cause unintended consequences. One of the major controversies surrounding synthetic biology is the ethical implications of creating life. Synthetic biology allows scientists to manipulate the genetic material of organisms and create novel life forms that do not exist in nature. This raises questions about the role of humans in shaping life and the ethics of playing God. Some people believe that the creation of life is morally wrong and that synthetic biology is playing with fire. Another controversy is the safety of synthetic biology. The creation of novel organisms raises concerns about the potential for these organisms to escape from the lab and cause harm to the environment and human health. There is also the potential for these organisms to be used for malicious purposes, such as bioterrorism. Environmental concerns are also a major issue with synthetic biology. The release of genetically modified organisms into the environment can have unpredictable and potentially harmful consequences. There is also the possibility that these organisms could spread and outcompete native species, disrupting ecosystems and altering the balance of nature. The commercialization of synthetic biology has also been a source of controversy. Companies are racing to patent and commercialize synthetic biology products, leading to concerns about the privatization of life and the potential for monopolies on new life forms. There are also concerns about the exploitation of developing countries, where synthetic biology products may be produced with fewer regulations and oversight. Although the field requires significant investment and resources, leading to concerns about the unequal distribution of benefits and the potential for synthetic biology to exacerbate existing inequalities. The discovery of synthetic biology will forever be remembered as a turning point in the history of science and technology. A moment when the boundaries of what was once thought possible were pushed back and a new era of discovery and innovation began. Today, it remains one of the most exciting and rapidly growing fields in science, with the potential to revolutionize our world in countless ways.